India want to become the knowledge economy. And to be that, stress on STEM is very, very important.
I want to know what research project currently you are working on.
We are working on two different aspects. The other one which we are working on is so if we can block it, we can kill the bacteria. Water molecule also need transporter to get inside the cell. So the phosphate transporter is an essential molecule, because phosphate is involved in working at an institution like JNU or UCSF. Not only teach you the science, but also this gave me enough courage to take these kind of challenges. It was the first membrane protein structure from this country. The first thing is the student must be good enough. He was awarded Nobel Prize for solving another membrane protein. So the list is long and it's growing.
Today on Genetic Education Podcast, Raw and Real, we have a scientist who is shaping the future of medical science. We have Dr. Hemant Kumar, sir. So, welcome.
Hello, Tushar.
Yeah. Dr. Kumar has received prestigious Ramalingaswamy Fellowship and is currently working as a professor of medical science at Charoset, Gujarat. With over 30 different publications in leading international journals, his work significantly contributed to our understanding of human health and diseases. Once again, sir, welcome and thank you so much for accepting our invitation. We are truly honored to have you here with us in this discussion.
Thank you, Tushar. It is good to talk to you regarding science in India and science overall, because India want to become the knowledge economy. And to be that, stress on STEM is very, very important for upcoming generation.
True, sir. So let's begin our discussion with something simple yet foundational. We want to first know, personally, I want to know what research project currently you are working on.
So we are working on two different aspects. One is related to small molecule discovery, that is against the infectious disease. The other one which we are working on is antibody engineering and antibody discovery. So small molecule discovery, actually what we do, we isolate compounds or we synthesize compounds based on some clue from our own research or from other research, and then we test it on target microbes. If we found it good, then we move forward and do the detailed analysis of those compounds. So small molecules means the molecules which are less than 1 kD, 1000 Dalton. So this is the small molecule one. The other one is antibody engineering or antibody discovery. These days, with like traditional medicine or small molecule drugs, a boom is there about the antibody, or you can say the drugs which are part of human system. So antibody are the natural defender of our system. In cancer, it is a name is there, which is called immunotherapy, which is replacing the traditional chemotherapy and radiation therapy. It is with help of these antibodies, monoclonal antibodies. So we are trying to find out novel antibodies, and we want to see what is their effect, and if there is a potential either for therapy or for diagnostics.
So it is about to provide knowledge for treatment, diagnostic, and therapies.
Yes.
Okay, sir. So I noted that your work involved transporter proteins. So what does transporter mean in biological context? And what is the significance of that transporter proteins?
So transport is a basic phenomenon and it is needed for all kind of biological process. Simply saying, transport is something which is out of the cell. It comes inside the cell with this phenomenon, and to facilitate this phenomenon, the molecules involved are called transporters, ion channels, and sometimes receptors. Though receptors are not in true sense a transporters, those transport only signal, not the substance. So whatever you think, small molecule like water or large molecule like big proteins. Whatever you think, it goes inside the cell through this process. At the same time, all the waste material, it comes out of the cell from the same process, means the transport. I can give you some example which should explain this thing. There is one protein which is called CFTR. CFTR is
Hmm.
cystic fibrosis transmembrane
Membrane regulatory protein.
conductance regulator. So this CFTR is a chloride channel, chloride transporter. So it transports chloride from outside to inside the cell. These proteins are essential in nature. So the modification in these proteins may cause disease, and sometimes these modification could be lethal. So like in CFTR, if the protein is modified, the transport could not take place properly, and that becomes the genesis of cystic fibrosis. Similarly, only in the human, but these transporters are important in all other genera. So, for example, MMPL3, this is a transporter present on membrane of mycobacteria, and this is essential protein for mycobacteria. If this protein is blocked, or if this protein somehow have some mutation which may block it, then mycobacterium dies. So in those microorganisms, these transport protein, though these are essential for bacteria, for us these are the very good drug target. So if we can block it, we can kill the bacteria. Here I would like to say that these transporter transport not only essential molecule but some non-essential molecule as well. If you think of a water molecule, water molecule also need transporter to get inside the cell. Generally, when the aquaporins were discovered, it was thought before that water doesn't need any kind of transporter. It can cross the membrane without any facilitator. But the discovery of aquaporin told us that water also need transporters.
So in case if that transporter like mutate or if it cannot work, that means our cells cannot carry water. Does that
Yes,
mean so?
yes.
In
And
that case, yes, sir, continue.
yes, and that cause problem means a very well-known problem associated with aquaporin is the deficiency of aquaporin zero, or sorry, modification or mutation of the aquaporin zero, which is present in our eyes. And if that happens, the eye may easily get cataract, glaucoma, all such kind of thing, dryness of eyes, all such kind of thing happen.
Right. So transporters are, in simple language, we can say very important molecules for every cells because it transports the nutrients and so many other things that a cell require. And on the other side, it also transport the waste from the cell. Interesting,
Yes.
sir. Now move on to our next question. We want to know your work on LacY. You have contributed substantial work, research work on LacY. So we want to know that experiment, I mean that research, and what is the significance of that particular research work.
So LacY, it was the LacY, which is called lactose permease. Late Professor Ronald Kabak, he was the pioneer in that research. So, and he has the authority on lactose permease. Lactose permease is a protein in the lac operon, which was, discovered by Jacob and Monod, for which they received Nobel Prize. So it is one of the protein in that lac operon. And what it does, it transports lactose from outside of the cell to inside of the cell. This LacY, which is also standard model of membrane proteins, because many structures of this protein has been sought. Not all other membrane protein have so much different structures as the LacY. So this LacY is from E. coli. So what is the importance of this, we all, as an infant, we take milk, right? And milk mainly contain lactose in the form of sugar. So during the infancy, the development of our gut microbiota depend on the supply of these fuels like lactose. So it helps our gut to get the proper microbiota and microbiome. And at the same time, it is a survival strategy for those microbes. In absence of sugar, it can survive using other sugar. And also, let me just quickly wrap up this LacY thing. This is a 409 amino acid long protein, and this actively transport lactose from outside of the cell to inside of the cell. And then that lactose further degrade into glucose and galactose, and then finally the galactose also changes to glucose and join the normal catalytic pathway, which is glycolysis and the Krebs cycle, and in that way it produces the energy. In the LacY, I think I should also mention another transporter which I worked on. It was the phosphate transporter. So phosphate transporter we worked on, it was from one of the fungus. And when we worked on that transporter, it was the first eukaryotic transporter of that category, which was solved structurally, atomic structure of first eukaryotic membrane protein that transporter has been solved by us, which was this phosphate transporter. So the phosphate transporter, phosphate, it is an essential molecule because phosphate is involved in many, many processes. You say energy production in the form of ATP, DNA production that also need phosphate, many signaling process that also need phosphate. So phosphate is an essential molecule. If you stop the supply of the phosphate, the cells cannot grow. It is also important in cancer as well. In cancer cells, two things are important. One is glucose, the other one is phosphate because they are rapidly dividing. So the rapidly dividing cell need both the energy and the backbone structures. So these phosphate transporter are very important. That's why the cell have multiple phosphate transporter. So it is called redundancy. So they have redundant pathway to acquire phosphate. So this phosphate transporter was amazing to see for us that when we say phosphate, so phosphate is triple negative ion. So phosphate cannot cross the membrane in that form because it is highly charged molecule. So the structure told us that how that phosphate binds to this transporter and then move on the other side. So this was also very important finding. After we solved the first this transporter, the method we adopted, through that method, many other eukaryotic human transporter of the same family were solved. It happens only after our work.
So, sir, what I understand is if we can somehow block in case of cancer, if we somehow block the phosphate transport
Mhm.
protein, we can say, so there might be chances that we can overcome cancer cells, or we can prevent the progression of cancer. Is it possible?
It should be selective enough, which can only target the cancer cell. But
Okay.
that is little difficult because phosphate is essential for all the cells. So such
Hmm.
an selective inhibitor means that is needed to be looked at.
So it's friend and enemy, I guess, both.
Yeah, sure. It happens
Interesting,
with
sir.
all the things. Yeah.
Interesting. Okay, so you work at JNU and world's prestigious medical school, which is UCSF. So how did that shape your research journey? I mean, how you evolved as a scientist?
Actually, working at an institution like JNU or UCSF not only teach you the science, but also the ethics of the science, also the passion of the science. So I don't like say that I only acquired the scientific acumen being in these big institutions, but I also learned how the process of science is a democratic way of looking at things. Because if you don't give importance to all the possible aspects, you cannot reveal the truth. So the institutions shaped overall me as a person, not only my scientific aspect. Like when I was working at JNU, this project was given to me that I have to solve the structure of phosphate transporter. At that time, nobody in our country, it was 2006, no one in our country was working on structural biology of membrane protein. So I could not find anyone to which I can go and seek some suggestions, some guidance. Even my supervisor, he was very enthusiastic to solve it because he had some initial discoveries related to that transporter. But being at JNU, I could have the courage to take that problem because you can work with big people who have done something big that give you that courage that, okay, you can also do it. So this gave me enough courage to take these kind of challenges. So when we solved the structure of this phosphate transporter, it was the first membrane protein structure from this country, from India. And then based on that, I moved to UCSF because this work was done in collaboration with UCSF. So I moved to UCSF and there I took it further. I further worked on lactose permease and there I worked with Professor Robert Stroud and Professor Ron Kebat. UCSF is a big institution. In UCSF, I was working at Genentech Hall. The Genentech Hall, I was working on fourth floor. Floor one, the ground floor was the facility. The first floor, second floor, and third floor. So in US, you don't have ground floor. It is first, second, third, fourth, right? So second, third, and fourth, all the three floors had at least one Nobel Prize winner. It was a very good environment, and in the other building, which is Gladstonian Institute, I think, I remember, two or three Nobel Prize winners were there. So it was a huge impact on my thinking on doing research, how to, even like when you do research, you learn how to record the thing. Because this is very important, which we many times forget in our life. We don't record the things. We might have very good observation, but we forget to write it, forget to record it. So
Hmm.
these institutions shaped overall many things.
Inspirational, sir. You achieved so many things like from JNU, which is indeed a very prestigious place, to the UCSF. So, first thing I noted that we have to, as a young scientist, we have to take note of everything that is noted. But what do you advise more? Like 3 or 5 tips to young scientists so that they can do better for them and for the country and for the world and for the science.
So let us take one by one. If you say advice to young student, the first thing is you should have believe on yourself, because if you don't believe on yourself, then even the smaller task becomes big. So this is one thing. Second, the student must be a science student, especially, although it apply to all of us. The student must be good in observation, because all the things you see in the science, they started with an observation. I just give you a small example. Raman effect. Raman effect was discovered by an observation by Professor C.V. Raman when he was going, I think, to UK or USA. On the deck of the ship, he saw the sky, and in the evening, the sky was red. So that observation led to the question. So the third thing comes, questioning. So a student must be able to ask questions. So he questioned why the sky is red, and then to find the answer, he discovered Raman effect. Similarly, observation and question, I can tell you another inspiring story of discovery of, I think many students or many people know about it. the discovery of smallpox vaccine. The smallpox vaccine, it was discovered by, means it was started by an observation. Edward Jenner observed that the cowherds, the people who have cow for their livelihood, they have cow. So those people don't get smallpox infection very easily. When he observed that, he asked the question, why it is so? And to answer this question, he discovered the vaccine for smallpox and vaccination itself. So asking question, and then the next thing which I would say is be methodical. As I said, write down, note down. So if you ask the question, you have 5 different answer in your mind, note it down. And then last thing is analyze the thing without any bias. Don't bias yourself that this cannot happen because you don't know what happened. I can tell you one example with my own experience. When we were working on phosphate transporter, the size of the phosphate transporter or any transporter which crosses the membrane should not be more than, I think, 40 angstrom, because the bilayer thickness is roughly 40 angstrom or maybe 20 angstrom, right? So membrane protein require detergents to purify those membrane proteins. The detergent which we use for purification of membrane protein, they have a head group and then a carbon chain. Similarly, like you see lipid, they have a head group and then carbon chain. At that time in the lab, people were saying, and this was common, people were thinking that any detergent which is having the length less than the thickness of the membrane, it will not work with the membrane protein either for purification or for crystallization. The protein will precipitate. When I was working, we had a problem that we were getting a good purified protein, but we were not getting crystals. The same dilemma came that, okay, we cannot use detergent which is having length less than the membrane thickness. What I thought, let us give a try all different kinds of detergent. And to my surprise and to everyone's surprise, nonide glucoside, which is having 9 carbon and a head group of glucose, it worked best. We get protein crystallized. It diffracted to 2.8 angstrom. So it gave all the good result, and then this was replicated in many more protein. The same nonyl glucoside was able to crystallize many more protein. So this was like, if I would have been with a religious thought that, okay, this thickness, the size of the detergent of membrane thickness is the best one and nothing other will work, then I could have not achieved this. So one should be away from the bias.
So this is truly inspirational, sir. This discussion is not just limited to science, what we have discussed. If I want to extract from this podcast is that it is about a journey of doing science and inspiration that you gave to young scientists what to do, how to do, self-belief that will inspire many peers and scientists in upcoming time.
Tushar, for inspiration thing, I just want to take 1 minute.
Sure, sir.
This area, the field of membrane protein, especially membrane protein structural biology, it was very decorative field. Many Nobel Prizes were awarded for working on the membrane protein, especially deducing the structure. Starting from 1988 with the Hartmut Mitchell, who received Nobel Prize for solving the structure of bacteriorhodopsin. Then came to 2003 when Peter Agre was awarded for discovering the structure of aquaporin. In 2008, Rod McKinnon was awarded for solving the structure of potassium channel. Then comes 2012 when Brian Kobilka and another associate, they were awarded to solve the structure of GPCRs. And in 2013, again, it was awarded for transport of vesicles. And I think recently, Julius, he's from UCSF only. Uh, Professor Julius, I forgot the last name. He was awarded Nobel Prize for solving the another membrane protein. So the list is long and it's growing.
The list is long, stories are also endless. But for this session, we are wrapping now. So thank you so much, sir. Thank you for giving us time and your valuable research insight and discussing so many stories with us, and future generation scientists will get inspiration from your stories and will do better for our country, for science and for themselves. Thank you so much, sir.
Thank you, Tushar. Thank you.